Hey, hello guys, welcome, vanakkam, vandanam, namaskaram. Today we are going to see molecular basis of inheritance part 3. So last video we have discussed a lot and the before video also we have discussed a lot. And this is a very important video where we are going to see replication, transcription and translation. Okay, so yeah. Replication. First, we will just check about the central dogma process, even though I've written it a lot of times where it is here. Okay. So first, what is this central dogma process? By this process only, all the characteristic traits that we have, I can say, I don't know whether no hormones will not count in every enzyme. Okay, every enzyme. For an example, if I'm having this color, it is because melatonin is secreted in this particular limit. Okay, so that particular limit is being controlled by the gene. Okay, see, we all know that this is a chromosome. This is also a chromosome, right? Okay, so in this chromosome, there will be gene. I'm going to erase it, so that is why I'm not writing this in a very beautiful way. Normally, my handwriting will be very beautiful. So, yeah, this is also very beautiful, right? Okay, then inside this, there will be DNA. Wow, very beautiful. Okay, so DNA is there, right? So DNA will be going first. It will be doing replication. Then from replication, it will be doing transcription. From transcription, it will be doing. It will be converted into RNA. Then RNA will be converted into proteins by translation, and these proteins are responsible for the characteristic expressions. Okay, so this is what the central dogma process is. This lungi is very comfortable. Okay, so coming to central dogma process, first of all, this replication is the thing that we are going to discuss, right? So this was found by Watson and Crick. Okay, so this was found by Watson and Crick in the year of 1953. Okay, so in the year of 1953 is the year that where these two persons have done. These two persons have found about this one. Okay, so in the year of 1952 only X-ray diffraction method had taken place, and also a lot of still another research has also taken place in the year of 1952 only. Those a separate 5 minutes video will be put for this lesson alone, since there are a lot of scientists. We will be discussing about all of them. Okay, scientists, the year and what they have found. In that video, these years and name of scientists will be very easy for us. Okay. So yeah, first of all, you have to know about this replication. See, for an example, this is what a DNA is, right? Okay, so this is how a DNA is going to be. So in this, I'm just going to consider this much more. Okay, so this DNA will be first, what it will be doing is, it will be in order to replicate, what it will be doing is, first of all, this DNA, the DNA like this is there, right? Okay, so it will find a particular location and it will undwindle. If this is what the tanglement is, then the tanglement will be untangled. Here it is untangled. So here, this is how it is, right? So here, in this place, new DNA will be growing. The new strand of a DNA will be growing, but one thing that you have to notice, okay, one thing that you have to note is that the direction of these two strands will be opposite to each other. For an example, we all know that in this, if this is a 2 strand, for an example, I'll take that this is actually, okay, so this is actually 5 dash. And if I consider this as 3 dash of this top strand, then for the bottom strand will be 3 dash, and here will be 5 dash, right? Okay. Then for this new strand, how it will be? For this new strand of the bottom strand, it will be 5 dash, and then here it will be 3 dash. Okay, and the bottom strand is there, right? So for this strand, there is the new replicating strand is there, right? So for that one, it will be 3 dash, and then here it will be 5 dash. So one thing that I have to say is, the new strand is being created, right? That particular strand will be always created from 5 dash to 3 dash only. Always, it is a golden rule that you have to remember. That is, this new strand. That the new strand, okay, the new strand will be always created from 5 dash to 3 dash. Okay, so the new strand which is being created in this DNA will be always created from 5 dash to 3 dash. That is how it will be always happening. Okay, so this is a very important rule. So from this now you might get some doubt. I will clarify all of them. Okay, so now I'm going to take this diagram in a very good way. Some people pronounce this as dash, some people pronounce it as prime. Okay, so it's completely based on your it's completely based on your wish. It can be said as 5 prime, or it can be said as 3 prime, or it can be said as dash. Your wish. Okay, see, this is how the normal DNA will be. Since I'm a very good artist, I have drawn in such a neat way, you have to appreciate me. Okay, so this is actually, so for this strand if you're talking about, so this is the strand that we have taken. This is the strand that we have taken, right? So this is for the top strand. I'm going to say this as the top strand. So this top strand, this end is actually 5 dash. Then for the bottom strand, automatically this will be 3 dash. We know now, we have already studied in our previous videos. Okay, so here it is going to be 3 dash. That means for the top strand, here it is 5 dash, then it is going to be 3 dash. It is like this: 5 dash, 3 dash, 3 dash, 5 dash. Okay, or 5 prime also you can say. Okay, so this is how it is going to be. Now the new strand that is being created, one will be created in this direction. Okay, and another will be created in this direction. Both the new strands will be opposite in direction. If one is being created like this, then another will be created like this. Okay, wait. So now this is what being created. So this is 3 dash. So automatically we know that this is going to be 5 dash. This is, since it is 3 dash, so this is going to be 5 dash. Okay, so it's just going, and it will be ended in the 3 dash. What is our golden rule from 5 dash? The new strand has started from 5 dash and then it has completed with 3 dash. So this is what the golden rule is. Here also see, so here it will be creating, so here is, this is what the direction is. Like this only it will be going. So here for this bottom strand, what it is going to happen here is where 5 dash is going to start. Okay, so like that it is going, and then here it will be ending at 3 dash. Okay, it will be ending at 3 dash. So this is how it's going to be. I hope it's all clear. So this is what the replication process is. So this particular process where two different strands are being formed, right? So we always know that when the replication process happens, then automatically the Chargaff rule will be followed by DNA. Okay, so what is that? Always adenine will be binding with thymine, cytosine with guanine. Like that only it will be consequently bonding. So that one we know, right? So that is how it will be happening. So this particular process is known as semi-conservation. Okay, semi-conservative DNA replication. This sort of DNA replication is known as semi-conservative DNA replication. Okay, so semi-conservative DNA replication. Okay, so then automatic, so this is, I hope the first paragraph has got completed. Next is the experimental proof. Now, we know that it's being created like this, but there is no such sort of evidence in that time. Now we have a lot of evidence, but in the year of 1953, when Watson and Crick found this, we did not have all these experimental proofs. They said that. So now we are going to see how did they find. Okay, now we are going to see how did they find. Okay, so it's going to be condensed here. Okay, next is the experimental proof. So what is that experimental proof? See, this is actually found by the same two persons only. Those two persons only did that experiment, and then only they found, they actually figured it out. So first of all, what they did is they considered a vessel. Okay, so in this they have put E. coli. E. coli is, I think it's very abundant. That is why every scientist is taking E. coli in the okay. E. coli had been taken. So for an example, if this is what E. coli cell is, then this E. coli cell will be having DNA in it, right? So like this only it is going to be, let us consider. Okay, so like this only it is going to be. Okay, so this is what the DNA is. Okay, so this is what the DNA is. So in this one, if you just so we know that DNA is having a lot of substances in it, like it has a nucleotide, right? So nucleotide and then some other bonds are there, phosphate is there. Nucleotide itself is bond with phosphate, so nucleotide has phosphate in it. So all these things are there. So and this E. coli, it wants a medium. It wants a medium. Not only for E. coli, for any organism, it wants some sort of ecosphere to live. So like that, this ecosphere is, in this case, the ecosphere is going to be a nutrient medium. Okay, so it's going to be a nutrient medium. So this E. coli, it is not just one cell. They have taken some cells, okay, and then they have put in a medium where this medium is actually having. So this medium is actually having what? It has heavy nitrogen in it. Heavy nitrogen. Heavy nitrogen in sense what? Heavy nitrogen in sense it is N15, an isotope of nitrogen. It is not radioactive, it is just heavy. Okay, it is not radioactive. It can be asked as one more question that you have to note that it is not radioactive. Okay, it is not radioactive. So this E. coli is not radioactive. So this is having heavy nitrogen in it, and we know that any DNA has nitrogen bases in it, right? It has some other portions also in it. So this one is going to replicate. We always know, see, note this one point here on it, that is for replication, resources will be used. Okay, surrounding resources will be used. Okay, so not only for replication, for any process, for any metabolism of an organism, absolutely the surrounding resources only will be used, right? For an example, for digestion we want the food that is from the surrounding. Okay, so for respiration we want the air that is in the surrounding, right? So for any process we want the resources from the surrounding, like that only this E. coli is going to consider this one. Okay, so it's going to split up. Okay, it's going to split up. So since I cannot draw those many times, I'm just going to take only one DNA strand and I'm going to erase together. Okay, so now if you just consider the diagram, it's going to, now I will be saying, see, this is normal DNA. So okay, normal DNA that is not having this N nitrogen 15. That means before putting this E. coli into this nutrient, the DNA is going to be, okay, the DNA is going to be drawn in white color. Okay, DNA is going to be drawn in white color. So for the differentiation purpose, what I'm going to do is the DNA which is being replicated inside the nutrient of E. coli will be drawn in orange color. Okay, will be drawn in orange color. Now let us check it out. So first one DNA molecule is there. Okay, this is before putting. Okay, so it's going to split. It's going to split, right? So this one DNA molecule will be converted into what? Okay, it is going to be split into two different strands, and each strand will be having another this strand will be developing its own strand, and this strand will be also developing its own strand, but where these two strands will be having that nutrient, okay, that's special DNA, the DNA which has nitrogen 15 in it. Okay, so this is how it is going to be. Now what I've done is, again, okay, so from this, Okay, so from this I am taking it out, and then again I am putting it into a normal ecosystem. Okay, normal ecosystem I am putting it. So in that case, so in case if we are putting it in a normal ecosystem, then how it will be? Okay, so this is how it is going to be, where this top strand is here, right? This is actually normal DNA, this strand is also normal DNA, whereas this strand is not normal DNA. It is actually a DNA which is also N15 in it. Also this DNA is also going to be having N15 in it. No, hereafter only these two strands will be hybrid strands. Okay, only these two DNAs will be hybrid DNAs. Okay, even if the next step happens by replication, then also there will be only two DNA hybrids will be there. Only two hybrid DNAs will be there. No matter now hereafter how many hybrids are there, no matter how many hybrids, there will be only two different hybrids are there. Okay, so why has he done like this? Okay, first he has taken it. Okay, then he has put into some sort of nutrient. Okay, which has N15 and other substances which is important for the replication of DNA. And again, why has he transformed or transposed these to the normal medium? What is the use of it as a normal question? Very simple. Wait, before that, we are going to write. This is in normal condition. Normal condition. Okay, and also this one. Okay, so this particular medium is not only having N15, also some other resources are there. Okay, plus essential replicating resources. Essential replicating resources are there, right? So this is in normal condition, and then next this one is actually in which condition it is? It is in the this condition. Okay, N15 condition. You'll write it as N15 condition. Again, when it is being transformed to the normal condition, then it is like this. Okay. If you just check it out, that I'm very elaborately explaining this one video, because this is very important and I should not forget in my lifetime, and also you should be understanding this one video a lot. So that is why I'm stressing out. For this one video alone, it will be like this. Okay. So see, this is taken, this is also taken, and this is also taken. Each had been put into three different, not test tubes, centrifugal tubes. Okay, and it had been centrifugated thoroughly. Then see, by using CSCl. Okay, by using CSCl. No, here I'm going to draw those centrifugal tubes. Here I'll write. Okay, by using CSCl, which is cesium chloride. Okay, gradient. Okay, gradient. They have used CSCl as its gradient, and then they have centrifugated it. Then what will you be getting? Oh God. Okay, so for some reason it is not looking like a centrifugal tube, it is looking like some other thing. Okay. So when he centrifugated it, he got only one. Okay, only one. So this one, when it is being centrifugated, he got this one strand. Okay, this one strand. Once again. Oops. Okay. So well, what he did is he did not just take these one. Okay, these one. change is here. Okay, one change is here. As I said that he used this heavy nitrogen, then he had grown a big colony. Okay, he had grown a very big colony where with this E. coli. Okay, so after that colony, from that colony he had taken some E. coli. And you have to note that in that 90%, okay, in this medium itself, in this medium even he has grown a big colony. In that literally 98%, not 98%, except two strands, all the strand of E. coli will be having heavy nitrogen only. It will be having one and only heavy nitrogen. It will be not like this. Instead of that, okay, so instead of being like this, it will be like this. Only heavy nitrogen will be there. There only that normal DNA strand will not be there. Normal DNA strand will not be there. So here only nitrogen 15. Here it is hetero, or instead of hetero I will write hybrid nitrogen 15. Okay, hybrid nitrogen 15 DNAs. Here it is actually 1N only nitrogen 15s. Okay, will be there. Okay, so now you are actually centrifugating it. I'm so sorry. Okay, so yeah. So when it is being centrifugated, then you will be getting a thin line in the bottom of this tube. Okay, the bottom of this tube, which represents that this particular DNA is having one and only N15. Since it is very heavy, it will be in the bottom. Since it is very heavy, it will be in the bottom due to gravitational force. Actually, it will not be like this. It will be horizontal. But since in the book it had been given like in the vertical, actually it will not be like this. It will be actually vertical. But since in the book it is given like in the horizontal way, so I have taken it like this itself. So this is actually having only N15 in it. I will write that later. Okay, so when you are centrifugating it, what you will be getting? You will be getting two different lines. One line is in the bottom. Okay, and then there is also another line that is not in the bottom, that is in the center of the tube. Not in the center, somewhere above this bottom line. Okay, so like this it will be. When you are considering this one, okay, when you are considering this one, it will be having like wait, it will not be this much far away. This will be a little bit here. Okay, these two will be adjoined with each other. Yeah, so these two will be adjoined with each other. Okay, so when you are centrifugating this one, you will be getting the line which is little bit. First, you will be getting these two strands, which will be very in the before itself. In the pre or even you will be getting this one in the latter. Okay, and later you will be getting these two, which is in this part. Okay, so like this you will be getting. If you just take it out, this will be having only N15, this will be having N14, and then this will be having N15, this will be having N14, this will be having N14, this will be also having N15. Okay, so this is what, if you want you can take a screenshot. Okay, so then after now we are going to check it about again back to the process. Okay, we are going to know. See, this is what 1 equalize. This is what 1 equalizer is. Okay, so this equalizer is there, right? So this 1 equalizer is there, right? So this 1 equalizer will be split into 2 different equalizers. Okay, so I'm just saying that this replication process of equalizer. Okay, so this it will be taking around 20 minutes. 20 minutes to split into 2 different equalizers. Okay, so in this one, if you're just taking it out, why am I using it? If you are just taking it out, oh, it's not even that much fancy sentence. Okay, so 18 minutes will be taken for what? For DNA replication itself. For this DNA replication itself will be taking around 18 minutes, and remaining 2 minutes only cytokinesis happens. Okay, other than that, for remaining 18 minutes, karyokinesis is what happening. This lungi is very comfortable. Okay, so karyokinesis. Remaining 2 minutes, only cytokinesis is happening. Okay. The common question is. The common question is, if you're just checking out the rate, before that, I have to say one thing. The DNA which is in it, okay, the DNA which is there, right, it will be like this. Okay, it won't be like this. Like this, it won't be. Very fancy diagram, right? Thank you. I will take it as a compliment. Okay, so since you all have to get some basic, not some basic idea, one important idea about this circular DNA, I am showing it over here. Okay, like this only it is going to be. Well, I can edit it, but I don't have the patience to edit it all. So just look it out. Like this only it is going to be. My phone is having 51% charge, but I am not even having 20% of charge. Okay, so karyokinesis will be happening for 18 minutes and 20 minutes. But one important thing that you have to check is that the NCRT data, you have to check it out. It is correct data only, but many people will be getting confusion that the rate they have given. Okay, so rate that they have said that about 2000 minutes. Okay, 2000 base pairs per second would have got formed. Okay, 2000 base pairs per second would have got formed. But when we are calculating, you will not be getting this one. You will be getting 4000. Okay, you will be getting 4000. Okay, you will be getting actually 4000 because rate is there. Now we are actually going to check about it. Since you will just go in everywhere, in all around. Okay, so what's happening is rate. So about 4.6 into 10 to the power of 6 base pairs are there, and if you are just checking out the timing, you will be getting 18 minutes into 60 seconds, so that it will be converted into seconds up. Okay, so when you are simplifying this one, you will be getting 4260 base pairs you will be getting. Okay, this is what the rate you will be getting, but approximately which can be taken as 4000 base pairs you will be getting. Okay, 4000 base pairs you will be getting. So in this one, okay, so in this one, this is actually for, I said this is what the structure is. So this is how the structure is. I'm a very good artist, so that is why I'm able to draw this good. Okay, okay. See, so in this one, this is totally, if you're just taking it out, totally there will be 2000 base pairs. Okay, 2000 base pairs will be, sorry, 4000 base pairs will be there. 4000 base pairs will be there, including this one and in this one. Okay, so this one and in this one, there will be totally 4000. But when you are taking only one, then you will be having 2000 only. So they are saying only for one. Okay, only for one, not for these two. Okay, So yeah, so you have to divide it with 2. You have to divide it with 2. That means you'll be getting 2000 base pairs per second. Okay, per second you'll be getting all these. Okay, so now after this, now we are going to check about replication. Here what we have seen, sorry, we have just completed replication. Now we are going to see transcription. Many people find that it is actually kind of hard, it is kind of tough. So yeah, transcription we are going to see. Along with transcription also the mechanism of the enzymes also we are going to see. So every enzyme and also transcription we are going to see. Transcription and unit, that one. Okay, so first we will complete this transcription. Mechanism. Okay, so the machinery and the and the enzymes. Machinery. The enzymes. So machinery and then the enzymes. Okay, so first we are going to check about ORI. We are going to check about ORI. I'm just going to talk in this. I'm just going to write and then I'm going to write the full form. Then some we will be writing functions. ORI is the origin of replication. I will write in a different color so that will be very good. Okay, so I'm going to use this color. Okay, so for an example, we are going to take this one chromosome. You just imagine. Sorry, this one DNA. You just imagine. Okay, in some sort of place, this DNA has to undwindle itself. That means it has to split, right? So it can be in any place. So, but it has to be in some place. So that is determined. That particular point, that particular origin is actually determined by this ORI only. Okay, it is what the initiator is. Well, there is a separate concept of promoter, terminator, or all, but it is not like that. It is just the initiator. It is just the location. That's it. Okay. Okay, so ORI is actually the origin of replication, where this is the point where you can actually say that the DNA transcription begins. Okay. Sorry, DNA replication begins. Sorry, this is all about replication, nothing about transcription. Still, we haven't even touched any transcription. Okay, so ORI. Then after this ORI, there is this another. Okay, there is this another thing that is called as helicase. Okay, helicase is there. That a helicase is actually unwinding. It will be unwindling. Okay, it will be undwindling. Undwindling in sense, see, if you are considering this as what it is, so this is what the point of this is what the point where it should be separated. That means again it will be so this is the point where it has got separated up. So in this is the point where ORI has chosen. Okay, ORI has chosen. Okay, so here, which means that this is actually unbundling. This is the point where ORI has chosen, and action is done by helicase in this point which is unbundling. That's the thing. Okay. So this is helicase. Then after with this one, next is topoisomerase is there. Okay, or gyrase is there. Both are same only. Topoisomerase or gyrase is involved in the unknotting. For example, if there is a tangle, okay, if there is a tangle, see, very simple. If you're just taking a thread, okay, if you are just taking a thread where the thread is being rolled like this, automatically in some places it's very common that we will be getting knots. Okay, wow. It's actually, I was thinking to get a thread, but I'm kind of feeling lazy to take it out. Okay, so yeah, it will be automatically making some knots, yes or no. Okay, so here I have taken two different threads. Okay, so this is one thread and then this is also another thread. Okay, now it will be actually forming this helical structure. Okay, so from this one I'm just taking one strand out. See, automatically a knot is being formed over here. A knot is being formed, right? So in order to not to get such sorts of knots, or even if we get a knot like this, now then what will be happening in DNA? Now then in that place this replication will be stopped, but this should be happening within fraction of seconds. Replication, transcription, and oh, I'm very close, right? Sorry, replication, transcription, translation, all should be happening within seconds. Okay, within fraction of seconds. So if in case a knot like this happens in sense, then it will be very problematic. It will be really very problematic. So in order to remove this knot, there is this topoisomerase. Okay, so in order to remove this knot, there is topoisomerase, or which is also known as gyrase. To remove tangles. Okay, then after this topoisomerase we are having primase. Okay, so what is this primase? See, okay, now it has removed it. Now the DNA which is like this, like this, now it has become, right? So here ORI should be, sorry, here polymerase, DNA polymerase is an enzyme which is very important. DNA polymerase 1 is there, DNA polymerase 2 is there, DNA polymerase 3 is there. Like that RNA polymerase 1 is there, RNA polymerase 2 is there, and RNA polymerase 3 is there. Okay, like that there are a few enzymes which are responsible for DNA polymerization and then also RNA polymerization. So here RNA, so here DNA is right. So DNA polymerization, for DNA polymerization, what should be happening is this enzyme, that DNA polymerization, that enzyme, okay, should be coming and it should be attached to this DNA. So to the point of attachment, here primase is there. Okay, primase is what? It is the place where this DNA polymerase acts. The place where DNA polymerase 3 acts. Really, I'm saying DNA polymerase 3 is the fastest, accurate, very accurate, very precise, very fast. So after this, we are having here some other protein also. We are having here single strand binding protein. Single strand binding protein is there. So it is actually, see, now it has got split like this. Okay, it has got split like this, right? Again, it should not be attached. Again, it should not form that helical structure. So to prevent that double helical structure, in order to prevent the reformation of double helical structure, we are using this, not we are, that it is using single strand binding protein. Okay, so to avoid the strands to get back together. Okay, to get back. Okay, so after this, what we are having? We are having sliding clamp. Okay, so there is this sliding clamp where this particular is the right way. I'll be showing that also. I'm not going to show you the sliding clamp. I don't know whether we can see it with our naked eyes or not. But one thing I can show, that's once again. Okay. See, this is how the normally DNA will be looking. Wait. I will be showing you that one shots which I had seen. Just to look at it. I hope I don't get any copyrights. This is what the DNA polymerase is, okay, and this is what the DNA is. It's just going, okay, it is just going. Oh, okay, so yeah. Okay, see, this is what the DNA polymerase is, that brown color thing, and then there is this this DNA molecule is, so it's going, and then it is going to attach to that DNA molecule. So after being attached, just see how it slides, how fastly it slides and how fastly it creates the new DNA. Just check it out, how amazing is it? So since it is this much amazing, so it should not go, see, for an example, that DNA polymerase should not just slide off and it should not fly off like that. Since it's moving in such a speed, it cannot just fly it off, right? So for that one, so to slide, to clamp it out, okay, to anchor it out, we are using this sliding clamp to anchor the what DNA polymerase 3. Okay, so before this sliding clamp itself, we will be having the action of, see, it is being written in a sequence. First RNA, next helicase, next topoisomerase, agarase, next primase, next single strand binding protein. Then next we will be having DNA polymerase. Polymerase 3 will be acting out, and after that one we will be getting sliding clamps. Okay, so here coming back to this topic, sliding clamps is there. So this, yeah, I've said about the sliding clamp, right? Okay, so this will be creating a new DNA molecules. Okay, see, next you will be having out this RNA molecule. Sorry, RNA materials. Sorry, raw materials. Raw materials. So raw materials will be formed. Raw materials in sense, these raw materials are important for the formation of the new DNA. For an example, if DNA is there, then DNA is a long chain of nucleotides, okay, polymer of nucleotides, right? So those nucleotides will be formed. Okay, here the raw material itself, those nucleotide is what the raw material. And the energy for the purpose of attachment. For an example, if this is what the strand is, this is how the DNA is. Okay, now it will be splitting, like this will be going, right? Okay, like this will be going, so now the new attachment, now a new strand will be formed here and here, right? So this new strand will be exactly complementary to this strand, and then this strand will be exactly complementary to this strand, right? So it has to, the nucleotides will be attaching with each other, and then for that some sort of energy is required. Okay, some sort of energy is required. For that, what we'll be using is, we'll be using TPs in sense ATP, GTP, like that we are having all those TPs, right? ATP will be used, GTP will be used, CTP will be used, TTP also will be used. Before that, deoxy, you have to put the word called as deoxy, ATP, deoxy. Like that, see, how many are there? ATP, GTP, CTP, and TTPs are there. For the attachment of ATP to this new strand, we want, to this old strand, to attach this, see, this is for the new strand and this is for the old strand, right? So this new strand has to be created from this old strand only. So if this is what the old strand is, so for this old strand, to this old strand, for example, if this is the nucleotide is, this is, let us consider this is adenine, okay? So this is what that old strand is. So to this, a new strand has to be formed like this. So for that one, another nucleotide has to come and join, right? So to join into this this nucleotide itself, there should be some amount of energy that is required. Okay, so that energy, for an example, if the joining is actually ATP, then there will be ATPase. Okay, ATP. So not AS, just ATP. Okay, so there will be ATP. If in case the joining is guanine, then it will be GTP. If the joining is cytosine, it will be CTP. If the joining is TTP, then it will be thymine, then it will be TTP. Okay, ma'am. Perfect. Okay, now we are just going to check out the structures. Just any one structure we are going to check. Okay, so for an example, I will be just taking this as the nucleotide. Okay, let us just consider this as a nucleotide. Okay, where there will be sugar, there will be nitrogen base, and then there will be a phosphate, right? So along with this phosphate, there will be still two more phosphates, those will be attached. So since this is actually 3-phosphate, what is the full form of ATP adenosine triphosphate? So here also it is actually triphosphate only. This is actually triphosphate. So like this only, in this place of nitrogen base, if there is adenine, so this is ATP. If in case if there is G, then it is GTP. If in case there is C, then this is CTP. If in case this is T, then this is TTP. Okay, so yeah, next after this, the DNA polymerase is there, right? What we'll be doing is, if a DNA is like this, then it will be first not DNA polymerase, it is other jobs. Okay, so it will be like this. First DNA will be like this, it will be dwindled, then it will be taken out, then it will be like this, then the new DNA will be formed. So there is a particular structure, called as replication fork. Okay, see, okay, so first I'm going to give the nomenclature. If this is 5 prime, then this will be 3 prime. If in case this is 5 prime, then this will be 3 prime. Okay, if it is 5 now, here it will be 3 prime. If here it is 3 prime, then here it will be 5 prime. Okay, so in this I'm just going to consider this one strand. So if I'm considering this one strand, from 3 prime, it will be going like this. Okay, from 3 prime to 5 prime, it will be going. Okay, from 3 prime to 5 prime, it will be going. There won't be any breaks in this DNA molecule. Okay, so in this DNA molecule, there won't be any breaks. It will be just growing on. Okay, so this is how it actually happens. Okay, so this strand, okay, this is actually known as continuous synthesis. Okay, so this one in this place, if you're just taking it out, I'm just saying, if you're just taking it out, note, you should not be used taking it. Okay, so from here it starts. Okay, from here it starts. Okay, so like this only it actually starts, and here it is known as discontinuous synthesis. Discontinuous synthesis. Okay, and there is newly synthesized strands. Okay, all these are newly synthesized strands, also known as Okazaki fragments. Okazaki fragments. Okay, what are they? They are actually known as Okazaki fragments. Okay. Yeah, that's it. So in this, if you're just checking, not again checking it out. No checking it out here at this. Here, this is known as, these two are known as template. Okay, template DNA strands. Template DNA strands. Okay, so template DNA strands is what their name is. Yeah. So again, now since these are all fragments, then they'll be all attached. They'll all be attached. That's how it's actually happening. So this is actually known as replication fork. And also the golden rule is, again I'll be writing the golden rule, no problem. From 5' to 3' only will be formed. Always from 5' to 3' only the new strand will be formed. Now you can get a confusion. Okay, already I've cleared it, it's okay. For this one, for this DNA, it is 5'. No problem. This is 5', here this is what 3'. From here only it starts. Okay, so for this Okazaki, it is what 5', it is what 3' is. Okay, always remember this. Okay, ma'am. Okay. After this is transcription. Okay, so transcription. Only one strand is copied in the process of transcription, not two strands are copied. This is the first point that you have to remember. This is what the wait. Okay, so this is what the DNA is. Okay, this is what the DNA is. So in this is actually I've drawn big, right? Okay, so here RNA polymerase. Okay, from transcription we know that RNA only will be produced. Okay, so from transcription RNA will be produced. That means if this is what the DNA is, so this is DNA, then from this DNA. Okay, so now the DNA has got replicated. So in that replicated, two DNAs will be formed. So in that, if you're just taking out only one, not checking. Okay, so if you're just taking out one DNA in that, okay, this is what the DNA is. So here I've considered this one point. Okay, this one point. So here is the place where RNA polymerase 3 will be acting. Okay, so template strand. Okay, so RNA polymerase 3 will be acting. This is actually known as RNA polymerase 3. RNA polymerase 3 will be acting out in this particular point. Okay, now coming to this case. Here there are two different strands. Okay, I'm going to give their names respectively. That it is known as. See here, this is there now. So this is actually said as 3 prime. This is actually known as 5 prime. Okay, so this one I'm talking about. This is what it is. Okay, so this is going to be 5 prime, and then this is going to be 3 prime. 3 dash also you can say. Since my biology teacher says it as prime, and it's a very good to think. That's kind of comfortable to hear, so I'm using prime. Okay, prime video. By the way, comment what series you are watching in or what movie you are watching in prime videos. Okay, so coming to here, I'm watching Two Broke Girls. Very good laughing thing. Okay, so I'm going to consider one and only this top strand. Okay, so in this top strand, what is there? Here is the place where RNA will be copied from this DNA, from this strand only RNA will be copied out. Okay, ma'am. So yeah. Like this, it will be copied out. So this copied RNA is known as hnRNA. hnRNA, new RNA, it is actually known as heteroRNA, okay, heterozygous RNA. Heterogeneous RNA, sorry. Okay, so this top strand is there, right? This top strand is known as template strand. Template strand. Okay, and then this bottom strand is there. This is known as coding strand. Okay, so from this only RNA is formed. Okay, and also this is known as non-coding strand. Okay, so for an example, this DNA is there, right? So this DNA is formula, or this DNA is nucleotide formula, we can say this as template. See, if it is AT, I'm going to erase it, so that is why not right. Okay, I will have it. So it is ATGC, again ATGC, like this it will be going on, and this is what I'm going to consider it as. This is what is going to be considered as this template strand. Okay. Oh god, time is running out. Okay, so this one is actually considered as, okay, so exactly complementary, but the thing is, instead of adenine you have to put only uracil. Okay, so thymine, uracil, cytosine, guanine, thymine, uracil, cytosine, guanine, thymine. Oh god, thank god current hasn't gone. Okay, so thymine, uracil, cytosine, guanine, because in RNA, this is what the RNA is. Okay, this is what the newly formed HNRNA. Okay, yes. So to this, I think. Okay, so to this newly standard HNRNA theory, that this will be exactly this one. Okay, this newly formed HNRNA will be exactly similar to. Will be exactly similar to the coding strand, because see, one and only difference is thymine, adenine you have to put in place of uracil, then cytosine, guanine, thymine, adenine, cytosine, guanine, thymine, adenine, cytosine, guanine. Like that will be going on. Okay, this is just known as, yeah, this is known as coding strand. Okay, so this is known as coding strand. Okay, just compare these two, HNRNA and coding strands. Their one and only difference is that here uracil is here and here adenine is there. Okay, that is what the one and only difference is. Okay, so yeah, so coding strand, DNA dependent RNA polymerase, template strand, all these we have seen. Okay, now one thing that I have to say is only one will be copied. Only from this, only it will be copied. Why not from this? If in case two are being copied, now first of all there will be double helical RNA will be formed. Okay, double helical RNA will be formed. Or if in case double helical RNA is not formed, only two different RNA even is formed, we can take it out. Okay, but those two different RNAs will be completely opposite in characters. For an example, if this RNA is doing something, let us do that. Let us take that it is actually doing some X sort of job. Then from this also, if in case RNA is being copied out, now then this will be doing X inverse job, exactly opposite job to this one. So it can form a mud. Okay, it can just form, not mud in sense, it can be toxic, or it can be something, it will be giving us some side effects. So that is the reason why only one RNA is being copied out. Okay, so then later on, yeah, okay, there are a lot of problems me taking video. It's okay, so I'll continue, no problem, no matter what. Okay, there is this one particular enzyme which is actually known as DNA dependent RNA polymerase. DNA dependent RNA polymerase. Here it is not just said as RNA polymerase. Okay, here it is not just said as RNA polymerase, it is said as DNA dependent RNA polymerase. Okay, so DNA dependent RNA polymerase, it is because the RNA which is being formed is there, right? It is completely dependent upon this DNA. If in case the DNA's nature is changed, then automatically this RNA's nature will be also formed. So that is why this RNA is completely dependent upon the DNA. Okay, so it is like this. I'm not ending the video because of my face. I don't take it wrongly. Okay, so there is also a reverse transcription that we have to see, but we are not going to check everything. Just I'm going to see that the enzyme's name. Okay, just the enzyme's name. Okay, so reverse transcription is done by germinism. Okay, germinism. Reverse transcription is done by Geminism. Okay, so done by germinism. Germinism in sense, see reverse transcription, it is an enzyme, okay, it is an enzyme which is used by the viruses, where viruses are having only RNA, right? So that RNA will be transformed into DNA, and in this DNA will be input into, it will be given to the host, okay? So that will be taking this DNA and it will be replicating, and then total immune system will be gone away, then Markaya Sala, okay, Markaya Sala only. Yeah, so reverse transcription, it is done by germinism by viruses. Okay, by viruses. Okay, so after this we are also having the okay, so see transcription unit. Transcription unit. If this is what one DNA is, okay, if this is what the DNA is, so then there will be two different things that you have to notice. Here one factor will be there, here also one factor will be there. Okay, here also one factor will be there. Okay, here sigma here rho. Okay, density and then sigma is used for what in physics? Okay, I forgot. Okay, so sigma and then rho will be there. Okay, so here this sigma, okay, this is actually located. The sigma factor is there, right? It is actually known as promoter. Here it is known as terminator. Okay, so still 5 minutes is there. Okay, so here this is the place where RNA polymerase. Okay, so this is the place where RNA polymerase okay, RNA, I'll write in blue color. RNA polymerase 3. Then after that also alpha, beta, beta dash, gamma. Okay, these all are known as core enzymes. And when also to this core enzyme sigma is known, sigma is also added, then it is actually known as holo enzymes, also known as holo enzymes. Okay, so all these are there, right? Actually, see, if this is what the strand is, here is where sigma factor is. Okay, so this sigma factor is the factor that is actually adding this RNA polymerase, this alpha, this beta prime, and then this gamma to this strand. Okay, to this strand, that this sigma factor is what adding there, so that RNA, H and RNA can be formed. Okay, so this is what responsible. The sigma is there, right? That is what responsible for initiation Okay, initiation, then also for elongation. Okay, for these two things, this is what actually is responsible. Okay, so yeah, this sigma is actually what the promoter is, and this is what the terminator is, and then this remaining region is known as creston. Okay, it actually can be said as creston. Okay, cystron. Okay, also known as structural gene. Structural gene. Okay, so when you are adding this promoter or this creston, or also known as structural gene, promoter plus creston or also known as structural gene plus terminator, then it gives you the what is that, transcriptional unit. Okay, it gives you the transcriptional unit. Transcriptional unit. Okay, also this gives you the transcription unit. Okay, so coming to the sigma, it is actually located in two different places. In eukaryotes, it is located in a place, and in prokaryotes, it is located in another place. Okay, it is located in another place. Okay, see, coming to prokaryotes, it is located in Tata box. Tata box. In eukaryotes, it is located at Tata box. Okay, this Tata box is also known as Pribnow box. Okay, Pribnow box. Pribnow box only. Pribnow box, also known as Pribnow box. Okay. In this, you have to check that this particular thing is there, right? So this is known as template strand. Template strand, and then this is known as coding strand. Oh. Okay, so with this one we will be ending this video, and then we also have translation. Well, that's not in this part. Okay, this hnRNA is there. About this hnRNA, once again I will be just saying this hnRNA. This hnRNA will be converted into mRNA. Oh God. Okay, will be converted into mRNA by a few processes. Okay, so yeah, so when you are coming to those few processes, they are actually splicing, capping, okay, and also tailing.